right, so we're talking about mitosis in this video, and you should remember that mitosis is what cells use for regular cellular processes. So they're going to use this to replicate themselves, and whenever cells are needing to replicate, or I should say, cells might need to replicate in order for them to repair, or they might need to replicate in order to let the organism that they're a part of grow. So mitosis is for making new cells that are identical to the cells they came from. And meiosis, which we're not talking about in this video, but meiosis is for making sex cells. It's for making genetically different gametes. So let's get into this. Let's just draw a nice cellular membrane here. I'll make it a bit smaller, actually. We'll draw a cellular membrane here. And then inside of here, we've got the nuclear membrane. And we can add let's add some genetic material to it. Some red and some blue. So there's your cell, inside of which you have just your regular old genetic material. And you'll notice it doesn't look like you probably are kind of used to chromosomes looking like that characteristic X that you tend to see. But right now, the DNA is just inside the cell, loose in this form that's called chromatin. Chromatin. Let's make this a little bit more realistically sized. So this is how your DNA is usually in the cell, in this unspooled, loose form called chromatin. The next thing that's going to happen in interphase, so interphase is where we are now. I guess I should make that black. Next thing that happens in interphase is called growth 1, or G1. A bit better there. So in G1, the only thing that's going to happen is that your cell is going to grow. Work. All right, so G1 phase here, we can do G1 phase live. There you go. G1. The cell is just growing. It's increasing its size. That's the first part of mitosis. The next thing that's going to happen in mitosis is, here, let me do it like this. We'll go around the corner here. Is S phase. So during S phase, all that's going to happen is your DNA is going to replicate itself. There may be some other cellular processes happening as well, but your DNA is going to replicate itself, and I'm actually going to draw these little structures in here. So this little circle, this crappy little circle, let me try again. And actually, I'll put it in a different color. I'll put it in like bright yellow, so it's very apparent. This little circle here, so that little yellow thing there is called a centrosome, okay? I'll label it in black. So here's your centrosome. We're going to find out what that's all about later. But in S phase, your cell is not going to grow. It's going to stay the same size, but it is going to replicate the DNA. So it's going to just replicate, draw the same. This is the same cell, hasn't grown. Okay, and it's just going to take all the DNA that it has, and it's going to make a copy of it. So if you were able to see this DNA, if you were able to look at a little part of it, here's your original chromosome. Right? It's this chromatin form. It hasn't condensed yet, but we're going to draw it slightly condensed. It's going to replicate itself. All right. And when it replicates itself, it's still one chromosome. It's still considered one chromosome, but there are now two identical copies. And here, let's get the other one, replicate our other piece of DNA, our blue piece. So you still have two individual chromosomes. So you have one chromosome here, and you have one chromosome here. And the point that they're connected has a name. This spot right here has a name, the centromere. Move this on over here. So the point at which those two pieces of DNA are connected is called the centromere. And each one of those individual pieces of DNA, by the way, is a sister chromatid. So each of these copies, they're called two copies of the same DNA, is a sister chromatid. The point at which they're connected is referred to as a centromere, not to be confused with the centrosome. And you might not be able to tell right now, but actually inside this nucleus, twice the amount as you did up top. You have twice as much DNA down here as you did before. And also, you have another copy of your centrosome. So now you have sort of two centrosomes here. You have twice as much genetic information going on. All right, and that's because S phase has happened. All right, and let me clean this up just a bit and get rid of some of this information. So, you know, the two copies are called sister chromatids. The point at which they connect is called the centromere. All right, so now we have we don't have twice the genetic information. We actually have the same amount of genetic information that's just been copied. We have the same number of chromosomes, which in this organism's case is just 2. In human beings, you have 46 different, you 46 different chromosomes in your diploid form. The next thing that's going to happen in interphase is just another growth phase, okay? We call this G2. So here's G2, and as you may guess, the only thing that's really happening here is that the cell is just continuing to grow. Finally, okay. So the cell is growing, it's doing normal cellular processes, continuing to grow. It has, a copy, it's got its chromosomes copied, it's got those centrosomes copied, and at this point, the cell can actually enter into the mitotic phase. It can enter into prophase. So here, it can actually enter into prophase. It can go into mitosis. So if I were going to draw this as a cycle, which pretty much every book I've ever seen draws it like this, so I'll draw it like this for you. Your textbook will have it like this. You've got, we'll make G1 red. You've got G1, just the first part, okay? And remember, G1 is just regular cellular processes. It's growing, it's doing cell stuff. Then we have S phase, or synthesis phase, where it's copying its DNA, it's copying its centrosomes. Then we have G2, or the second growth phase, where the cell is getting bigger and doing just kind of regular cellular business. And then here we have the mitotic phase. This is mitosis. This is mitosis in here. And it's a relatively small part of the overall cell cycle. So it's just the part where it's sort of replicating itself. And you might be asking, well, what if there's a cell that doesn't necessarily need to copy itself? Well, it undergoes this thing here, which is called G0 phase. And in G0, it's just kind of hanging out. Maybe it's growing, it's making whatever products that it needs to make, but it's not doing a ton. So I went ahead and grabbed our cell from before and just put it back down here. This nuclear membrane, by the way, should be continuous. It shouldn't sort of be like open like that, but that's all right. So as we enter into the mitotic phase, or as we enter into mitosis, if we were to zoom in on our nucleus here, you would be able to see that these sister chromatids are now starting to look like what you would expect a chromosome to look like. They look like the typical that typical X shape because they have been replicated, and they are condensing. All right. So before they were uncondensed in the form of that chromatin, but now that we've entered prophase, they have actually condensed down into these actual chromosome-like looking things. And remember, the point at which they're connected, still called the centromere. And each of these is still one individual chromosome, right? And each of these pairs is called a sister chromatid. I don't know if I talked about that, but I'll just sister chromatid. I'm going to scratch that, actually. I'm going to make it a bit easier on you guys. We'll do this. Sister chromatid. Each of these is a sister chromatid. And again, during prophase, after G1S and G2, they've entered the mitotic phase, and the chromosomes are going to condense and start to look like you would expect chromosomes to look. These schematic X's here. All right, so what I'll do, just going to grab these guys. I'm going to kind of get rid of this stuff. And I'm just going to kind of cheesily, so they'll fit into our nucleus. That's cool. So during prophase, these chromosomes are condensing to look like the X's that you expect them to look like. The other thing that's happening here is that your nuclear membrane is starting to break down. So the nucleus of the cell, I'm just kind of whiting this out. The nucleus of the cell, the nuclear membrane, is beginning to break down, and these they are not centromeres, they are centrosomes, so be careful with that. I don't want anyone to get confused and think that something is a centromere when it's a centrosome, or vice versa. So these here centrosomes are going to migrate. They're going to go to the poles of the cell. I'm not really doing this justice, but they're going to start to migrate to the poles of the cell. And what we've just witnessed here with the chromosomes condensing, the centrosomes going out to the very edges of the cell, these things moving out here, that's stuff that all happens in prophase 1. Not prophase 1, I'm sorry, this is mitosis. This happens in prophase. There is no prophase 1. So again, prophase 1, what's happening is chromosomes are condensing, the nuclear membrane is breaking down, and those centrosomes are migrating out to the pole of the cell. They're going to the very extremes of the cell. So the next thing that's going to happen, a little arrow here, same exact cell, all right? You still have your centrosomes out here on the edges. And the next thing that's going to happen is your nuclear envelope is going to be completely gone, and your chromosomes, try to redraw it the best that I can, are going to line up in the middle of the cell. Okay, these things are going to line up in the very middle of the cell. Get this blue guy here. All right. So here, let me do this. We'll say here this was prophase. And then out here, oopsies, this is metaphase. All right, so when they line up in the center of the cell along this line called the metaphase plate, that is called metaphase. So there's a few more things happening in metaphase. So you have your centrosomes out here, and they are attaching to each other, as well as to a central structure inside this chromosome. Draw these a bit more evenly. Called the kinetochore. I'm not worried about the kinetochore right now. Just kind of know that those centrosomes are attaching to the chromosome. They're going to attach in the center. It's not really a centromere. It's a kinetochore, which is a protein structure associated with the centromere. But just know the centrosomes are attaching to these chromosomes. They're attaching to one another, and your sister chromatids, your set of sister chromatids, which together make up a chromosome, are lining up along the central line called the metaphase plate. And when you have those chromosomes lined up in the center, you have no more nuclear membrane. You have attachment to those centrosomes. You call that stage metaphase. So then the next thing that's going to happen in this process is that the sister chromatids are going to sort of separate from one another. Let me draw this real quick. So we'll put a centrosome here, the centrosome here. And you're going to have these little bits of DNA being pulled to either end. Remember, way back in S phase, the DNA copied itself. So you had an original piece of DNA, you had a copied piece of DNA, and those two together were called sister chromatids. You put them together, they made up a chromosome. And now when they start pulling apart again, now they are actually considered to be two individual chromosomes. So as they're pulling toward the edge of each cell, each of these things, here's one chromosome, here's another chromosome. Right? Those sister chromatids are now considered individual chromosomes. And the phase here where they're pulling out to either end of the cell, this is called anaphase. This is called anaphase. They're kind of messed up here. So really, in anaphase, should have drawn it like this to start with. It's okay. In anaphase, really your membrane here is starting to get pinched. It's just kind of starting to look a little bit like two cells. It's just the very beginnings of that process. Let me throw anaphase back here. So then the next thing that happens is a more exaggerated sort of pinch. You still have your centrosomes over here. I drew them smaller, but that was unintentional. They should be the same. And then you kind of got a chromosome here, and a chromosome here. You've got your other chromosome. These are starting to unwind, and you have the reformation of the nucleus is beginning. So the nucleus is starting to reform again. So this phase is called telophase. In telophase, the chromosomes are uncondensing, they're unspooling, and the nucleus, the nuclear membrane is starting to form again around that genetic material, around the DNA. So here's a better drawn version of all that I just borrowed from the internet. So over here you've got interphase, which again, the three phases of interphase are growth 1, where it's going normal cellular processes, it's growing, it's doing cell stuff, it's making protein. In the S phase, in which is replicating its DNA and those centrosomes. Then you move into prophase, which is where those chromosomes are condensing from their chromatin form, and you can see these two sister chromatids So each of those X's still represents a single chromosome, but it is 2 copies of that piece of DNA. Also in prophase, the nuclear membrane is breaking down, which is not shown here, but that is happening. The nuclear membrane is beginning to go away. This is late prophase. They also call this prometaphase, which I'm not as worried about. It's just showing you that it is indeed a cycle. And there are sort of steps in between each of these steps. Then in metaphase, you have your chromosomes lined up on the metaphase plate, and they are attached to those centrosomes. Next, in anaphase, the 2 sister chromatids are being pulled to either end of the cell, and they are considered to be individual chromosomes at this point. And then in telophase, you start to see this pinch of the cell, and you can see nuclear membrane is reforming. The nuclear membrane is reforming around both cells. And then finally in cytokinesis, the cellular membrane pinches off, and you have 2 individual cells here now that are theoretically identical copies of one another.